You all are very excited for the class that you come before the time. Yes, Ashrita, Medha, Geeta. It's like only my class or all the classes will be coming before the time.
Only your class.
Geeta, what about you?
What, ma'am?
My question is that you are there in the class before the time. I think somewhere 3 to 4 minutes you have joined it. So every classes you join like this before the time, or only my class will be joining before the time?
Ma'am, only your class.
Why not the others also?
I'll join exactly on time, but yours I'll join before time.
Okay. Yeah, okay.
Yeah, I joined 10 minutes prior.
Oh, okay.
I just joined after her by 1 minute.
Oh, and Medha, when you have joined?
Ma'am, like in what 25 or something.
Okay, 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 okay. Fine, that's good actually. Then we can start, soon. So I think Atharv has to join, Prahita, Rishit. Let them join, let them wait. We'll wait for a few minutes more because still we have the time. Okay, children, once now you can turn on the video. Let me see everybody. Okay, I can see Medha. I can see Ashrita now. What about the Atharv, Geeta? I can see Rishit now. I can see Geeta. Medha. Now I cannot see you, Medha. So in your school, children, don't you have the extra classes of kind of things and all?
Ma'am, my school will start since October, extra classes.
October month?
Yeah, not now.
Why a year before?
No, I mean like it will start on October this
Oh,
year.
it will start from October.
Yeah,
Okay, okay, okay. And Rishit, what about your school? They'll be giving extra classes kind of things?
No,
Okay, Atharv.
ma'am, they said they'll start it, but don't know. They didn't give the dates.
And Atharv, you stay in Madinagura?
Yes, ma'am.
I think your mother has joined that group, right? Madinagura, we are opening the branch.
Ma'am, don't know. I'll ask.
Okay, I'll talk to her. Geeta, for you, the extra classes will be there in the school?
No idea, ma'am.
Okay, Ashrita, for you?
Yes, ma'am, but this year not there. As they're renovating some things, it's not there this year.
Okay, okay. Fine, it's good if it is not there because then grade 9, I think it is not needed also. If their school are not keeping, that's okay. You can enjoy your life certainly. So,
Yeah.
okay. Yesterday we have discussed about the mitochondria, I guess. And today we will be discussing a little complicated topic called as the endoplasmic reticulum. Okay. So when we talk about the endoplasmic reticulum, any idea what it is? Atharvan, made turn on the video first of all. What is the endoplasmic reticulum? No idea, children. Any one of you have any idea about what is ER? What is endoplasmic reticulum?
Rough endoplasmic reticulum, smooth endoplasmic reticulum.
Okay, two types are there, rough and the smooth endoplasmic reticulum. But I'll tell you in the depth manner, what are they? What kind of the components do they have? What are the differences between RER? What are the three parts of the ER when we talk about endoplasmic reticulum? They have three parts also: cristae, vesicle, and tubules. And what are the functions? So, likewise, step by step, we will be learning the entire function with the parts also. Okay. And if in case you feel like it is much more and not able to understand, you can stop me. I'll just stop there. Okay. I'll try to explain in an easier way only, but still it might be like, ma'am, it is too much. And I don't want you to write anything, children, right now, okay? Because this topic is a little complicated. Just focus what I'm teaching, what I'm writing. And for writing part, maybe you can write after the class, or maybe I'll give you the time. It depends upon uh, completion of the topic. Okay. Hmm. Today is so soon the months are going on actually. Endoplasmic reticulum. I'll add the image first of all. Can you see, children, the structure of the endoplasmic reticulum, how it is?
Yes, ma'am.
Okay. So I'll explain you what are the different parts which are given here. See, this part is, sorry, this part is the nucleus. Okay. Mm, so this is the nucleus. As we have discussed already, the nucleus structure. Nucleus will have the nuclear membrane. Now, outside the nuclear membrane, as you can see on the nucleus, this endoplasmic reticulum, they are being attached. You can see in the picture itself. The blue color part, orange color part, they both are the endoplasmic reticulum only. Okay, they both are endoplasmic reticulum. Now, in this also, can you see the difference? This part has basically the small particles over them, which are called as a ribosome. And the part which is present here, they are they do not have the ribosomes on them. They are smooth. So that is how two types are there. Rough when they have the presence of the ribosome over them, and the smooth when they do not have the ribosome on the surface of them. So this rough endoplasmic reticulum, mostly they are attached to the nucleus only, but whereas the other type, which is the smooth, they will be present scattered here and there also. Okay. And uh, hmm. Another picture I'll show you, this one. Okay. Here, as you can see, three types are given, three parts I can say: cisternae, vesicle, and tubules. Now, cisternae, they are the this like structure. Okay. Vesicle are the round structure which are present like this. You can see here. These are what? They all are the vesicle. Then tubules are tube-like structure. So here in the center, you can see the tubes are present. So somewhere it is tube-like structure, somewhere they are circle-like structure, somewhere they are just simple sac-like structure. So that is how all the three types of the parts of the endoplasmic reticulum will be present in the RER as well as the SER. Talking about the points of it, endoplasmic reticulum is basically a large organelle. So what it is? It is a large organelle. As you can see in the picture itself, they are what? They are very large in the structure. So they are the large organelle that consists of network of membrane. So you can see in the picture, they are large at the same time. They are the network of the membrane. So many layers, so many membranes, so many network are connected together from each other. So ER is a large organelle that consists of a network of membranes. And also, what do they have? They have the tubular structure as well. So both the things they will have the tubular structure as well as the network of the membrane will be connected together. Now, be it the membrane, be it the tube, be it the spherical structure, all of them, what is their primary function? Their primary function includes the formation, packaging, and secretion of the cellular product. So, whose primary function, so the major primary functions will include the formation. So, whatsoever kind of the secretions are needed for the cell, they will be helping them to form, they will be helping them to pack, and they will be helping them to transport here and there. So, all those tasks will be done by the ER. Now, based on the presence or absence of the ribosome, based on presence or absence,
Ma'am,
yeah, tell me.
yeah, should we write with you?
No, don't write with me. I'll just complete it and I'll give you the time. Now, based on the presence or absence, the ER is further divided into rough endoplasmic. ER means endoplasmic reticulum. Rough and the smooth ER. As you know, rough, it is because of the presence of ribosome. Presence of ribosomes will be there on them. Whereas the smooth will have the absence of ribosome. This is the first point. Now, the second major difference for them is they are the protein factory. Geeta, can you tell me why they are the protein factory here now?
What, ma'am?
Why the rough endoplasmic reticulum, they are the protein factory?
Because they have ribosomes over them.
Correct. Since they have the ribosome, that is why these ribosome will be helping them to produce the protein. Whereas the smooth one, they are the lipid factories. Very, very important this is. So RER is for the protein formation, whereas the smooth are for the lipid formation in our body in the ER. Now let us discuss all the differences between RER and SER. RER and SER. The first major difference is that about the surface. In the rough, it will be rough. In the rough, it will be rough only. In the smooth, it will be smooth kind of surface. Second thing, appearance. How do they appear? So, rough endoplasmic reticulum, basically, they appear as like flattened sac. Like this, they will be appearing. A flat sac-like structure, they will be appearing. Whereas the SER will have the tubular network. They will have the tubes of the networks. Now, third thing, the main function, as I told you, keep in mind, They help, RER help in the protein synthesis, whereas SER help in the lipid synthesis. That is the next difference. Now, next one, location, where the RER and SER present. RER, as I told you, they will be attached to the nucleus, so they are present near the nucleus, whereas the SER, they will be more spreaded in the cytoplasm. So, they will be more spread in cytoplasm. Now, the next difference is detoxification. Atharv, do you have any idea what is detoxification? Atharv, Rishit?
No, ma'am.
No.
Ma'am, like removing the toxics.
What kind of toxics?
Ma'am, I just guessed it with the name. Ma'am.
Correct. Which is correct only, Rishit? Ashrita, any idea? Meda, any idea? Here, detoxification means, tell me.
Ma'am, it removes poisonous or harmful substances from some of substances like living organisms.
Correct, right. So detoxification means whatsoever kind of the drugs we are taking. Drugs does not mean the negative only drugs. It can be in the form of the medicines also. It can be in the form of the food also. Sometimes through the food also. Like for example, you have, the mithais will be there, the sweets will be there. Over the sweets, they will have those silver foil and all they'll be keeping, right? So once we eat them, then slowly and gradually those silver foil will go in our body and they also start creating the poison. So such kind of the poison, such kind of the drugs, the SER will detoxifying. Detoxifying means they will be removing the impact, they will be removing the effect of the poison from our body. So RER will not detoxify, whereas a smooth endoplasmic, the major work of them is that only that they help in the detoxification of the body. That is why the SER is also important. It's not only about the formation of the ribosome, it's also about the detoxification in our body. Then the next one, calcium storage. So in the RER, they do not require to store the calcium because they help only in the protein synthesis, so they do not need it. Whereas in the SER, they will be doing in the muscle cell. So for the detoxification, for the functioning of our cells and all, the storage of the calcium is required. That will be done by the SER only. Then the next difference is types of proteins and lipids. So basically RER, what do? They will make the proteins for the secretion of membrane. Whatsoever the membrane which get old or which get distorted, they will be forming that membrane with the help of RER. Since they have the protein, what they can do? They can generate the new membrane. And this process is called as biomembranogenesis. Biomembranogenesis, where the formation of the new membrane will be done with the help of the RER. Whereas the SER will make the lipids and also they will be making the phospholipids. So lipids are the molecules of the fats which is present in our body, and the SER will be helping them to make it. Whereas the phospholipids are the two components in which the phosphorus and the lipids both will be generated. Now, example here are the cells producing proteins like the plasma cells. So in our body, we have the plasma cells where the protein will be generated with the help of the RER. Whereas the SER, they are present in our liver cells and also they are present in our muscle cells. So these are some of the differences between the RER and SER.
Only no power of changes, no.
Medha, you can mute yourself. Medha, you can mute yourself. So rough, smooth, they will have flat structures, have bag-like structure. They will have the tubular structure. They help in the protein formation, whereas they will help for the lipid formation. They are present near the nucleus. They are present in the cytoplasm more in the much amount. The detoxification will not done by the RER, but by the SER. Calcium storage will not be done by the RER, but it will be done by the SER. Then they will help in the formation of the new membrane also, which is called as membrane biogenesis. And here the SER will help in the formation of the lipids and the phospholipids. The example which produce the RER or where they help it is the plasma cell, and in the SER they are present in the liver cell and the muscle cell. Am I clear, children, with this?
Yes.
Yes.
Any doubt, Medha, Ashrita, Athar, Rishit, Geeta?
No.
No, ma'am.
No, no.
Okay.
No, ma'am.
Then we'll go to this diagram only back. The first one here, what we are going to talk about is the cisternae. Cistern. Now these cisternae basically, if you see the structure, they will have a double layer. They are the two-layer structure. So they are two-layered and they are unbranched. If you see the structure, they do not have the branching. They are unbranched. They are thin component and also they are found close to the nucleus. Now, if I say they are found close to the nucleus, then what type of the ER they will have? Yes, children. If they are present close to the nucleus, then in the case of the cisternae, what type of the ER they will have? The rough one or the smooth one? Yes, Adar? Yes, Geeta, Rishit, Medha, Ashrita.
I'm the rough one.
Yes, they will have the rough one. As you can see in the picture also, right? They are like this flat structure and also they have the presence of small, small molecules, which are the ribosome molecule. So they will have the rough endoplasmic reticulum. So they are two-layered, unbranched. They are thin component which are present near to the nucleus. And these are joined, these cisternae, they are joined and they are stacked. Stacked means present one above the other. And stacked one on the top of other. So here you can see in the picture, this is the first, second, third, fourth, fifth. So what is happening? All are one above the other. Like if you have the coins, those coins, if you make it, keep it one above the other. Same way, these cisternae, they will be present in that form only. And they act as the main site for protein synthesis. So since they have lot of the ribosomes, they will help them in the formation of the protein synthesis. Then also they will help in the folding and modification in the RER. So whatever changes they require, those changes will be happening in the cisternae part of the ER. Am I clear, children, with this?
Yes, ma'am.
I can hear only Rishit's yes. What about the
Yes.
others? I can hear now, Geeta.
Yes, ma'am.
Atharv, I can hear now. Meda,
Yes.
Ashita.
Yes.
Again, I'm repeating, they are
Ma'am.
two layer, they are unbranched, they are thin, they are present near to the nucleus. Now, if they are present near to the nucleus, that means they have the ribosome. Now, if they have the ribosome, definitely they will be taking part in the protein formation. So, what do? They will help in the protein formation. They also help in the folding of the RER, and also they help in the changes which are being required in the RER. That will be done by the cisternae only. So in your book, they have not given about these things at all, but you can understand them. Then their size is around 40 to 50 mucometer in diameter. So that is about the cistern. Now, second one, they are the vesicle, which I told you, they are the round bodies like structure. So they are small, spherical sac. Sac means round bodies like structure that will be actually branched off, or it means it comes out from the ER. And what do? They help in carrying the chemicals to other organelle. In simple language, I can say that the main function they will be doing is to carry the substances from one side to the other side. So kind of transportation they will be doing it to the cell surface. And because of which they help in the transportation. So this help in transporting various cellular material. So whatsoever the material is needed by the cell, they will help them to get transferred, which includes lipids, protein. So whatsoever the lipids they generate, the SCR, and whatsoever the proteins, the RER generate, it has to move on actually. It cannot be at that place. It has to go to the other parts of the cell wherever it is needed, or other organ of the body wherever it is needed. So that work will be done by the vesicle. So they will help in the transportation of the various cellular material. Cellular spelling is wrong. Cellular. So they will be transporting the material like the lipids and the proteins to a specific sites within the cell. So wherever they need, they will be transferring within the cell, and then from the cell, they will be passing through the cell membrane, and from the cell membrane, it goes out. So like this, it will be taking place. And third one, they are the tubules. So tube-like structure. So they will be like this tube, which can have the branches like this. They can be like this. So they are the branches basically. So they are long. They are as well as cylindrical, long, cylindrical structure. Sorry. Long cylindrical structure that make up the part of the ER, part of the ER. And what do? They will provide the flexibility. And they provide flexibility. Flexibility to the ER. Now, why this flexibility is important? This flexibility is important because if the structure is flexible, then only the movement will be happening in the easier way. If it is hard, if it is tough, then movement will be difficult. That is why to make the flow easy, they are flexible. So they are the long tubules. They are cylindrical. They make the part of the ER, and also they provide the flexibility to the ER structure. And also, they are also involved in the, they are involved in the extension and connection of the ER throughout the cell. So they also help in making the connection. They will be acting like a bridge, taking from one place, giving to the other place, then taking from the other place, giving to the nucleus. So like this, they will be involving themselves as an extension and also a kind of the connection of ER throughout the cell. And they will also facilitate the movement of material. So whatsoever the movement of the material has to be done, they will allow them to move within the organelles. So vesicles, they will be transferring the proteins and the lipids within the cell. These tubules, they will be making the connection for the easily movement of the materials in and out of the cell. So that is how these three parts of the ER are working. Now, any doubt in this, children? Rishit, Geeta, Medha, Ashrita, Atha?
No, ma'am.
No, ma'am.
Now, the last one of this is the functions of ER. Now, one second, children. Hmm. 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 Is my screen visible?
Yes.
Yes, ma'am.
Okay. Now, the last one, they are the function of the ER. So, they will be doing it the flex. They will be giving the mechanical support. They give the mechanical support to the cytoplasm. Then they will also help in the formation of the proteins and lipids, as we have discussed, the RER and SER. Formation of proteins and lipids. Then they will be also helping in the transportation, as the tubules will be doing it. Transportation of material, they will be doing it. So this transportation of the material, especially the proteins, they can transfer it, especially proteins. They can transfer them from one part of the cell to another, or they can also do it between the cytoplasm and the nucleus, between the cytoplasm and nucleus. That is what they will be doing. And the SER, what they will be doing, they will be helping in detoxification. Detoxification. That means it converts the harmful poison and drugs into harmless substances. Into harmless substances. That is what is detoxification. Fine. So that's about the entire topic of the ER, children. If you want to ask anything, you can please ask that. Yes, Geeta, Athar, you wanted to ask anything? Medha, Ashrita, Rishit.
No, ma'am.
No.
No, ma'am.
Okay, then you can start writing now. And I hope children you are making one notebook only. You are not writing here and there anywhere whatsoever you feel like to do. Because if it is in
No.
one note, then it will be easy for you to actually revise. Start writing from here.
Mom,
Yeah.
what's the last word in the third line?
Last word in the packaging, secretion of numerous cellular products. It include the
No,
formation, pack where then?
I can see from since ear is a large organelle that consists of a network of membrane and the last word I
Tubular,
can
tubular,
tubular.
tubular structure.
Ma'am.
Hmm.
Ma'am, what's after based on the?
Presence. Actually, I've written the short way. Presence or absence.
Okay, ma'am. Done, ma'am.
Okay, everybody has done with this page?
Yes.
Yes.
Okay, children, then I'll send this file. You can complete and you can send it back to me. And today you have, I think, one more class, right? Like two more
Two
classes.
more.
Maths as well as the social is there. Okay, children, I'll send you in some time.
Ma'am,
Yes, Geeta, tell me.
ma'am, why aren't the class recordings visible in the app, ma'am?
Which class recording, which subject you wanted to have see?
Biology only, ma'am.
Oh, that is actually, since I'm taking it over the Zoom, it is not visible to you all. But whatsoever you need, you can message me. I'll provide it. Okay.
Okay, ma'am.
So whenever you need, we'll provide it to you.
Okay, ma'am.
Okay, Geeta.
Bye, ma'am. Thank you, ma'am.
Bye bye. Thank you.